Actions and properties of enzymes. How does enzyme work? Each enzyme has an active site. The shape of the active site is very specific. Reactions among the substrate take place within the active site. Enzymes bind to the substrate that fits the shape of an active site. In this case, the whole structure is called enzyme-substrate complex. Within the enzyme-substrate complex, activation energy is lowered. Reactions take place to form the enzyme product complex. In catabolic reactions, the substrates are broken down into products. The enzymes remains unchanged at the end of the reaction. In anabolic reactions, simple substrates fit into the active site of the enzyme to form enzyme-substrate complex. Product will be formed within the active site. The enzyme remains unchanged. Let's look at an animation of how enzymes work. This is an enzyme. Some substrates collide with the active site of the enzyme. In the active site, the substrates are perfectly held to form an enzyme-substrate complex. Reaction takes place. The substrate turns into product. The product is soon released from the enzyme product complex. The enzyme can be reused again. Types of enzyme reactions. In catabolic reactions, enzymes are used to break down complex molecules into simple molecules. Example, maltose can be broken down to form glucose. The enzyme is maltase. Many enzymes end up with the word ASE. In anabolic reaction, simple molecules are linked up together to form complex molecule with the help of enzyme. Example, excess glucose in our body will be converted to glycogen in the active site of enzyme. Glycogen will be stored in muscles and liver. What are the properties of enzymes? Enzymes are biological catalysts. They lower the activation energy to speed up chemical reactions. At the end of a chemical reaction, enzymes remain unchanged. They can be reused again. Therefore, to convert all the substrates into its products, only small amounts of enzyme is enough. The action of an enzyme is highly specific. Only those substrates with a particular shape can fit into the specific active site. For example, in lipase, the shape of the active sites cannot fit with glucose or protein, but it can fit with fat to produce enzyme-substrate complex. Only the enzyme with a specific active site fits a specific substrate, just like only the key with a specific shape fits the lock. This mode of action can be explained by the lock and key hypothesis, which is a scientific model used to explain that enzymes are highly specific in action. Enzyme in this case works like the key, and the substrate works like a lock. Only the key with a specific shape fits the lock. Enzymes are made up of protein. They are formed by polypeptide chain, coiled and fold up to form a protein which have specific shape. The specific shape of enzymes, or the active site, are held by bondings between amino acids. As enzymes are proteins, their structures are easily denatured at high temperatures and extreme pH. When we hit the enzyme vibrates more vigorously. At high temperature, the bonds which hold the enzyme in shape are broken. The three-dimensional shape or conformation of the active site changed. The enzyme denatured. Some enzymes catalyze reversible reactions. You have learned this example in Form 3. Carbonic anhydrase is an enzyme found in red blood cells. It plays a role in carbon dioxide transport. Inside our body cells, we carry out respiration. CO2 is continuously produced by body cells. CO2 will move into red blood cells. Inside the red blood cells, there is an enzyme called carbonic anhydrase. It will facilitate the reaction, turning CO2 and H2O into carbonic acid. When the red blood cell gets to the lungs, carbonic anhydrase will speed up the same reaction, but in opposite direction. That is, turning carbonic acid back to CO2 and H2O. CO2 will diffuse into the air sac. It will then be breathed out. Enzyme activities can be slowed down or even stopped by certain chemicals. Such chemicals are called inhibitors. This red object is an enzyme inhibitor. It changes the shape of the active site so that substrates cannot bind to it. Girls, can you tell me the properties of enzymes? Enzymes are biological catalysts. They speed up chemical reactions. They remain unchanged after the reaction so that they can be reused. To complete the whole reaction, a small amount of enzyme is required. The action of enzyme is highly specific. All enzymes are proteins. Their activities are affected by temperature and pH. Some enzymes catalyze reversible reactions. Inhibitors can slow down enzyme reactions. Thank you for watching. Goodbye.